Hello everyone and welcome to my YouTube channel. Today we are going to understand the protein sorting and transport. The first step of protein sorting takes place while translation is still in progress. So what is protein sorting and transport? Protein sorting or sorting and targeting is a biological mechanism by which protein are transported to their appropriate destination within or outside of the cell. Okay, so about 1/3 of cellular protein are processed in endoplasmic reticulum. Okay, so first we understand the endoplasmic reticulum structure. So the endoplasmic reticulum is a network of membrane enclosed tubules and sacs cisternae, okay, that extended from the nuclear membrane throughout the cytoplasm. Okay, suppose this is the nuclear membrane and nucleus. This is the nuclear membrane. Okay, and they extended throughout the cytoplasm up to this. Okay. The space enclosed by the ER, okay, ER, that means the lumen and cisternal space, may be represent about 10% of the total cell volume. And the entire ear enclosed by the continuous membrane and is largest organelles of most eukaryotic cells. Ribosomes are attached to the cytosolic phase of the ER membrane. So here it is the rough endoplasmic reticulums. Okay, this is the ribosome attached to the cytosolic phase, not a nucleus phases. Okay, outer side. The entire ear is enclosed by continuous membrane. Okay, this is a continuous membrane. Okay, so there are two connecting membrane domains within the ER that perform different functions within the cell. So first one is the rough ER, okay, which is covered by ribosome, as you know, on its outer surface or cytosolic surface, and its main function is protein processing. Now second one is the smooth ER, is not associated with the ribosome and is involved in lipid metabolism rather than protein. Okay, so endoplasmic reticulums and protein secretion. Okay, so the role of ER in protein processing and sorting was first demonstrated by the George Palade and his colleagues 1960s. Okay, so he conducted one of the experiment which name is the pulse chase experiment, and then he defined that pathways taken by secretory proteins. The secretory pathways is rough ER to Golgi to secretory vesicle and cell extracellular. That means protein are transported from the rough ER to Golgi, secretory vesicles and cell extracellular. He also demonstrated that mammalian cells, okay, most proteins are transferred into ER while they are being translated on membrane bound ribosomes. So this is the pulse experiment. Okay, the pulse experiment in which the pancreatic ansia cells, okay, we secrete most of their newly synthesized protein into digestive tract. They were labeled with radioactive amino acid to study the intracellular pathways taken by the secretory proteins. So first they label the amino acid and then incubate the cells for 3 minutes. So after 3-minute incubation with radioactive amino acid, it's called pulse. Autoradiography revealed that newly synthesized protein were localized to the rough ER. Okay, following further incubation with non-radioactive amino acid chase, protein were found to move from the ER to the Golgi body. Okay, so this is the radio label amino acid protein. Okay, then 7 minutes. Okay, they transport from the ER to the Golgi apparatus, and then within secretory vesicles. After 20 minutes incubation, change this protein from the Golgi apparatus to vesicles. This is the vesicles, transport vesicle to the plasma membrane and cell extracellular. Okay, so in high eukaryotic cells, okay, so protein sorting, okay, overview of protein sorting. In higher eukaryotic cells, the initial sorting of protein to the ER take place while translation is in progress. Okay, protein synthesized, okay, free ribosome cytosol. Okay, protein synthesized on free ribosome either remain in the cytosol, okay, suppose they remain, or are transported to the nucleus, mitochondria, chloroplast or peroxisome. But in contrast, okay, in contrast, protein synthesized on membrane bound ribosome. Okay, membrane bound ribosome, that means that ribosome are bounded with the ER membrane. Okay, this is the ER lumen, the cytosol side of ER, mRNA, okay, and protein. So this protein are translocated, okay, translocated directly to the ER through the translocan. Okay, this is the transmembrane protein, okay, translocan to the into the ER lumen. This protein contains signal sequence. This is the red color, it is the signal sequence, okay, indicated in the red, that are cleaved during the translocation. Protein here translocate into the ER, okay, translocate into the ER, may be either retained within the ER or they transport to the nuclear membrane. Okay, this is a nuclear membrane, not a nucleus. Okay, this is the protein are transported to the nuclear membrane, plasma membrane, secretory vesicles, endosome, lysosome and peroxisome membrane. Okay, so this is all about today videos. I hope this will help you. Thank you.